chloroplast genome is made up of a double-stranded circular DNA. Chloroplast genome is made up of the chloroplast genome is structurally made up of double-stranded circular DNA, and the chloroplast DNA has about 110 genes located on it. Okay, there are three most important regions present on the chloroplast DNA, and these three important regions are known as the inverted repeat region. There are two inverted repeat regions: right inverted repeat region and left inverted repeat region. Beside inverted repeat region, the other two important regions present on cpDNA include the SSC region, single short copy regions, and another is the LSC, long single copy region. So there are three major regions present on the chloroplast DNA, which are inverted repeat region, SSC region, and LSC region. In addition to these regions, there are some introns which are also present in the chloroplast DNA. You know that introns are generally present in eukaryotic genes, but the chloroplast genome, as we know that chloroplast and mitochondrial genomes have resemblance with the prokaryotic DNA, like the bacterial DNA, but there is presence of some introns also in the chloroplast genome, and there exists a special mechanism for the removal of the introns from the transcript, and there is complete absence of capping and polyadenylation, which are generally found in the eukaryotic eukaryotic RNA. As you know that eukaryotic DNA is at first transcribed into the hnRNA, and hnRNA is processed to form the mRNA, and the processing of hnRNA into the mRNA involves capping at the 5' end, polyadenylation at the 3' end, and the removal of the introns and joining of the exons. So this is true about the nuclear genomes, not about the organellar genomes. However, there is presence of introns in the chloroplast genome, and there is a special mechanism for the removal of the introns, but there is complete absence of capping and polyadenylation at the 3' polyadenylation. So this is altogether there are three very important regions present on the chloroplast DNA. You can say chloroplast genome, and these three regions are I have told you that these three regions include the inverted repeat region, single short copy regions, and single long copy regions, and some introns. So these are the regions present on the chloroplast DNA. As far the number of genes present on the chloroplast genome is concerned, there are about 110 genes, and there is very important features of the chloroplast DNA, that the chloroplast genome is completely devoid of, or is completely devoid of the D loop region. The mitochondrial genome has displacement loop, D loop, but the chloroplast DNA does not have the D loop, displacement loop, and the genes present and the chloroplast has got more genetic autonomy than the mitochondria. Okay? So these are the important features of the chloroplast genome. Now let us discuss the important genes present in the inverted repeat region, important genes present in the LSC regions, and important genes present in the SSC region. So let us at first talk about the inverted repeat regions. The inverted repeat regions has the genes meant for encoding the ribosomal RNA. You know that the chloroplast has its own ribosome, and for making ribosomal RNAs are required. So the gene for encoding ribosomal RNAs are present in the inverted repeat regions present on the chloroplast genome. And there are 4 types of ribosomal RNA present in the chloroplast, and these 4 types of the ribosomal RNAs are 16S RNA, 23S RNA, 4.5S RNA, and 5S RNA. So there are 4 types of ribosomal RNAs which are present in the chloroplast, which are required for making the ribosomes found in the chloroplast. And you all know that the cytoplasmic ribosome in the eukaryotic cell is of 80S type, but the ribosomes present in the mitochondria and chloroplast are of 70S type, like the prokaryotic ribosomes. So there are 4 types of ribosomal RNAs which are required for making the ribosomes present in the chloroplast. And these 4 types of the ribosomal RNA are 23S RNA, 16S RNA, 4.5S RNA, and 5S RNA. And for encoding these 4 types of ribosomal RNAs, the genes are located in the inverted repeat regions. Okay? So these are the genes for encoding ribosomal RNA are located in the inverted repeat region. Thus, other genes which are present in the inverted repeat regions are the genes for encoding the tRNA, transfer RNA. You all know that the chloroplast and mitochondria have their own infrastructure, like the ribosomes, like tRNA, like amino acid for protein synthesizing. So the tRNA will also be required. So there are different types of the tRNAs are present in the chloroplast, and for encoding these tRNAs, the genes are present in the inverted repeat regions. Okay, so the gene for encoding ribosomal RNA, the gene for encoding tRNAs are also present in the inverted repeat region. Beside this, the gene for encoding the RNA polymerase, you know that RNA polymerase is a key enzyme for catalyzing the process of transcription. So the gene for encoding RNA polymerase are also located in the inverted repeat region. Beside this, the proteins required for making the ribosomes, proteins are also required. You know that ribosome consists of ribosomal RNA and protein. So the gene for encoding the protein which will constitute the ribosomes are also present in the inverted repeat region. There are 3 genes for encoding RNA polymerase, and there are about 19 genes for encoding the ribosomal proteins which are located in the inverted repeat region. So these are the 4 categories of the very important genes which are located in the inverted repeat region for encoding the ribosomal RNA, tRNAs, RNA polymerase, and protein for making the ribosomal proteins for which protein, and the genes for encoding the protein which will constitute the ribosome in association with the ribosomal RNA. These are the 4 very important types of the gene located in the inverted repeat region. Okay, I have already written these 4 important genes on the whiteboard, the image of which has been shared in this group. You might have seen that. Now let us come to the LSC region. These 2 regions are very important. Inverted repeat regions and LSC region. Long single copy regions. You know that the chloroplast has granum, and the granum consists of a number of thylakoids present upon one another, present one above another. So granum, the thylakoids are made up of protein. So for encoding the thylakoid, the proteins are, for encoding thylakoid proteins, the genes are present in the LSA region. You know that the light reaction of the photosynthesis consists of 2 systems, PS1, pigment system 1, and pigment system 2, and the proteins and the genes for making the protein which constitute the pigment system 1 and pigment system 2 are also located in the LSC region. Beside this, you know that the ferredoxin, cytochrome, NADH dehydrogenase, for all these genes are located in the LSC region. That is you know that NADH dehydrogenase is a proteinase enzyme. You know that the ferredoxin is for a ferredoxin, and cytochrome oxidase, and cytochrome B, yes, you might have studied in, if you I think that you might be knowing all these cytochromes and ferredoxin and NADH dehydrogenase. if you have gone through the photosynthesis, do you know that the genes for encoding the proteins of the pigment system 1, pigment system 2, thylakoid proteins for encoding thylakoid proteins, cytochrome proteins, FD proteins, and NADH dehydrogenate proteins. For encoding such proteins, the genes are located in the LSC region. And as for the genes present in the SSC region are concerned, the genes are widely genes are arranged in the SSC regions, and there is a wide gap between the genes present in the SSC regions. And in addition to these regions, there is a presence of introns. As I have said earlier, introns are also present in the chloroplast genome. And these introns are removed with a special mechanism which is present in the chloroplast. So this is all about the structural organization of the chloroplast genome. In the next lecture,